Hello, so today I'm going to demonstrate how to extract, how to get DNA from strawberries, and hopefully I will success. And those are strawberries I bought from Sam's in Shanghai, and actually quite delicious. So I only use two for this lab. All right, I don't need to wash them, but I want to relieve to remove the leaves. Okay, so strawberries are plants, and in order to get DNA from plants cells, there are two barriers we need to to conquer, to went through. So first one is the cell wall, the what's it called, the cell membrane. All right, good. So how we do that, we can just put them into the bags and add some water. Okay, hopefully that's enough. And get up all the extra air. And okay, that's a little handy work. Get up the air, okay, and just smashing them up. Okay, so that's how I'm going to my strawberries. Okay, to break the cell wall. All right, so that's the first step. And what I have here is I have this baking soda and dishwash, and I have a glass. I also have a bottle of rubbing alcohol in my freezer, which I'm gonna use later. Okay, let me squish better. Okay. Is that too much water? I don't know. So this is my first time to try to do this experiment, and I'm really excited. Yeah, maybe a smoother will mix it better. I don't know. There's some still raw part of the strawberry. By the way, I think they're quite expensive now. It's like I think spend 90 RMB or something, and I only get 72 strawberries. Yeah, it's like you spend $12 and you have 72 strawberries. I think it's quite expensive. Okay, so I don't want you to wait longer, so maybe that's it. You still can see the raw strawberry. And okay, so well, the cells has been broken down, should be lighter, and the rest of the raw strawberry should be at the bottom. So I will try to kind of like a filter, okay, to make sure. Oh, sorry, just said filter, right? Let's filter it. Okay. Too close. Yeah, it's not an easy job. Okay. All right, here we go. Scoop. Squeeze. Oh no. Okay. All right. I just pull it. Yeah. A little more. I'm kind of out of patience a little bit. I'm such an impatient person. Squeeze it. Squeeze it. All right. Okay. Hopefully that works. All right. So that's the first step. Break the cell wall, and then we need to break the nucleus, so to get the DNA inside. And basically, the nucleus was surrounded by lipid basic stuff. So we use dishwash, and okay, let me try this. Okay, just a little, maybe a little more. Okay, yeah, very little, this much dishwash, and let's stir them out. Okay. So use this more basic dishwash to break down the lipid structure. Alright. And while I'm doing this, let me try to answer the questions regards to, okay, can we get DNA in those, this kind of process for other organisms? The answer is yes and no. So yes, you use kind of a similar process. The no is because, well, bacteria will be easier, but I don't have this much of a bacteria that I can, just use my eyes, use my kitchen stuff to put up the DNA. And also, the other stuff like seeds or fungi or meat, animal meat. If we want to get our DNA from those, well, they kind of have a more tough structure. So there's like a little bit, you need to modify this process a little, but pretty much the same, Besides animals and bacteria doesn't have cell wall, and that's one step. But again, they have a tough structure of their lipid, the other protein, the other surrounding and keep the shape structures are more tough. So you need to do something extra. All right. I don't know. Is this gonna work? Theoretically, what I said just theoretically. I never tried this before. This is fun. I think so. So hopefully, we use hands and bags and water to break down the cell wall, and now hopefully we broke the nucleus in a cell. All right. So let me put it down. Let's use our freezing alcohol. And the reason is that because, even though we get our DNA from the nucleus of the cell, but again, it's kind of so it's still a strawberry water solution. And remember, the great thing about water solution is that they have, they're using hydrogen bond. So that's what make water the best solution in the world. So basically, the hydrogen bond will connect with other substances like DNA. What do we have here? So we're using alcohol to separate them, so the DNA will stay together versus stay with the water. And let me try to put it carefully on the surface. Oh yeah, I don't know, but I think so. Okay. So, well, yeah, I think you can see this. So, yeah, so as you can see, it's kind of separated, and the downward side, the lower side was the strawberry water solution, and the upper side was the alcohol with DNA. Well, the reason is we can see them is because both DNA molecules and alcohol is actually less dense. Well, especially compared with the strawberries inside of the water solution. So they float up, and also because it's cold, I put it in the freezer. So a colder temperature will slow down the hydrogen bond moving with each other. So yeah, so the DNA molecules will stick together. Oh yeah, you can see more clear now. So yeah, so okay, so the cold temperature and the alcohol makes the lighter DNA molecules, make them stay together versus they stay with the other water molecules. And so they stay together, and because they're lighter, so they can try to flow up. And this is what we have. Yeah, I guess I know what DNA looks like. Right, and they try to answer the last question. So on this level, do we expect the other organism DNA looks the same or similar? Well, yes, because DNA is basically just it's just what it is. The full, I can't remember the names, but the full numbers, and they kind of like Lego. They kind of just stay together and make up different blocks. So on this level, yes, DNA will look all similar. The difference will just be some of the organism DNA will be longer, some of them will be shorter, and yeah, I guess yay, succeed with my 2 strawberries. See you later.